My name is Alika Kui. I am a student at Winter Community College in Microbiology 130 with Dr. Tina Michael. I'm here with two very special guests. Both have experience in biology. This is Amanda. She had a major in biology, and this is Cody, who actually did research. Can you guys elaborate a little more on your experience and backgrounds?
Well, I guess, yeah, I just graduated with my, I don't know, what's that called? My bachelor's in biology, and I just find it really interesting, so, yeah.
I too graduated with a degree in biology and public health.
Oh, cool.
Yeah.
From what I hear, Cody, you did research, right?
Yeah, I did in the cardiac center at UH's medical school.
What did you research?
I did a little bit of plate slide analysis of myocytes, which I would maintain cell cultures in the lab and make sure that the cultures were split properly and passaged, and make sure they're differentiated and all that fun stuff. Yeah.
Very interesting stuff. Very interesting stuff. So today's topic for my project, we have to talk about microbial growth and control, right? So from what I understand, all organisms, there's a few ways to control how organisms are in a lab, how you can control them, how you can monitor them. I guess because it's my project, I have to do most of the talking because they want to know if I'm understanding the source material. But you can correct me if I'm wrong, if I'm mistaken. This is an entry-level microbiology, so I don't know everything, but I'm learning. From what I understand, it looks like what most organism needs to survive is energy and carbon. Is that correct?
Yeah.
Usually.
Right.
So, in theory, I would think that if you take away one of those two things, energy or carbon, then they wouldn't be able to thrive, right? So I feel like that would be a good way to control it. And petri dishes, usually, that's where you do most of your research, right? Aren't those made of like sugars?
Yeah, it depends what kind of growth media you're using, I guess.
Oh.
Usually it's like agar plates.
Yeah.
But yeah, it's pretty cool how you set it up. It's kind of like a jelly.
That's I haven't taken the lab class yet, but I'm very excited.
Yeah. You can do smear plates and grow a bunch of colonies of bacteria.
Sounds fun. It sounds like so much fun. Another way to control, I would think, is most organisms, or the basic function of life from what I've been read in the book, is eat and procreate. So most bacteria, or all functions of life have these things, and the way penicillin works is it prevents the cell from, or bacteria from dividing, right? So if you can take away their ability to procreate, of course we just talked about the eating thing with the media, then you should be able to also control the growth of bacteria. Is that correct? You think so?
Yeah, that makes sense.
Yeah. Yeah, because antibiotics just disrupt one form of mechanism, whether it's producing proteins in the bacteria or inhibiting their defense. Yeah.
So then the question becomes, if all life has basic functionalities of what we need, and we're all like a collection of cells, how do you harm the bacteria that's infecting you as the host without harming the host? Does that make sense?
Let's answer that, Amanda.
I know. Can you repeat the question?
Well, that's why it's not. You're talking about like antibiotics, right?
Yes.
Because since they're not a benign drug, so that's why it's kind of like a broad spectrum that it hits the whole body. So that's why you have symptoms like diarrhea, like dizziness, headaches, fatigue.
So we can get as close as we want to harming our bacteria without harming us as a host. It's the little proteins that you need to go in and out of your membrane that I feel we target for bacterias. And think I hit all the points I need to for microbial control and growth. You guys are the experts in the field. Is there anything you guys would like to add?
No, I think
No,
you're an
that's
expert
cool.
now.
Yeah.
I think you're more of an expert than us.
Well, thank you so much, Cody. Thank you, Amanda. And thank you, Bree, my wonderful camera girl. Thank you. Thank you, guys.